we're plateauing off on people coming in. So today's talk is by Toby Kellogg. Thanks, Toby, for stepping in at short notice. And Toby will be talking about the genome that took over the world. And if you're ready, Toby, take it away.
Okay, I'll just, should I just go ahead and get started?
Yep, please go ahead. And you can post questions in chat or wait till the end.
Okay, I should say that this, because I sort of finalized this talk in fairly short order, I think it's too long. So I'm just going to go ahead and present, and at some point, I guess maybe 10 minutes before 11 or so, I will probably just stop rather abruptly. I don't know, do you usually have questions during the presentations for this, or do you just go through and ask at the end? I can
totally up to you. Usually people post questions in chat and then we can go through them at the end.
Okay, let's do that, given that the talk is already too long. And as I said, some people's stuff is probably just going to get chopped off. I didn't get it organized. So, right. Okay. So, in the last 20 million years, there's one group of grasses that has taken over 17% of the Earth's land surface. This is from a preprint by Caroline Lehman's group. And this is the tribe Andropoganae, which is shown on this map in orange. And you can see that it's pretty widespread and covers a lot of acreage. The name may or may not be familiar to you. It is named for the genus Andropogon. So andro means man and pogon means beard. And presumably that's referring to these very fluffy spikelets in that occur in many members of the tribe. The ending EAE, wait a minute. I should try to get the laser pointer. Is that the laser? Oops, that's not what I wanted. Can I go back? I'm trying to find the pointer. There we go. Okay, this ending EAE is the standard taxonomic ending for anything at the rank of tribe. And it gets pronounced differently depending on what your first language is. So whatever way you say it is fine. So anyway, Andropoganae, named for Andropogon. They are ecologically dominant, as you might have guessed from that previous map, and they're about 1,200 species. So this is about 1/10 of all the species of grasses. So it's a big group, but not enormous. And of course, as everybody on this call knows, in the last 10,000 years, a few members of that same group, Andropoganae, have become our most productive domesticated crops. And yeah, here's the maize, of course. And this maize is cultivated over most of the world. And what you can see just by looking at these maps is it's grown similar to spaces where a lot of the other 1,200 species of Andropoganae grow. So again, very widespread on many continents. And there's a problem in maize in that there's a lot of variation. And that in a few thousand lines, which raises a question of which alleles, which genes are really important for adaptation to particular environments and which are sort of incidental. And obviously you can't do all pairwise crosses to try to test that one by one. You'd like to know which alleles are deleterious and should be selected against in breeding programs. And dealing with those questions is just very hard looking at a single species and its genome. If you just look at maize, even with the enormous resources available, people have done tremendous work locating sequences that might be functionally important, open chromatin, MNase accessible sites, TF binding sites, etc. And then determining if they really are important is a lot of work. And then what they're important for is another question. Are there things that get missed by these various approaches? Are there regions of the genome that have been under natural selection? The big questions, challenging questions to answer if you're working in a single species, no matter how good the resources are. So the solution is to let evolution do the field trials and basically look at presumed regulatory sequences of the entire Andropoganae. That's, 20 million years, and then you sort of add up the length of all the branches. It's quite a lot of evolutionary time, and therefore natural selection captured by looking across the whole group. And looking for SNPs and presence-absence variants, regulatory sequences, and testing if they correlate with the environment or some other desirable trait that you're after. So basically the goal then of the project I'm describing is to take the genetic information from wild plants like this big blue stem on the left and to start thinking about which alleles or combinations of alleles matter for adaptation, and transfer the information to, not the genes, but transfer the information to maize, so that you can target particular things to be pursuing. So this was the sort of the motivation for the Pan-Andropogonii project, which I'll be talking about today. And the goals of the project are really very simple, given that overarching question. The first one is to sequence and assemble the genomes of every species in Andropogonii, align everything, and look for conservation and diversity in the sequences and correlate with the environment. So how is this done? Well, we managed to get some funding from the NSF for about 4 years, extended to 5. And this is the team, the Pan-Andropogonii team, led by Ed Buckler at Cornell and USDA, and involving Matt Hufford, Jeff Rossi-Barra, Adam Seipel, and me. So what did we need to make this happen? Well, first you need a whole lot of germplasm. And there's some descriptions of this and some beautiful pictures at this website that Taylor Aubuchon Elder in my lab put together. Need long read sequencing and good annotation of at least some of the species. And that good annotation means including the transposable elements, not just the gene space. For things where long read sequencing is hard for various reasons, there's short read sequencing and assembly, which at least will get the gene space and the orthogroups. And then there's a whole lot of tool development, including tools for improving the annotation of the maize genome, because if the annotation isn't good, then none of the rest of this works. Multi-species alignments of whole genomes and identification of regulatory sequences. And to make those tools work, a robust phylogeny is needed. So I'm going to go through these in order until I run out of time, although I'm going to spend most of the first part of the talk on the germplasm, because that was where my lab was involved. That was basically our component of this project. And it may be a part, if you're used to getting things from a stock center, then working with these wild plants is just a different experience, and you may not be familiar with all the aspects. So because I'm going to run out of time, let me give you the take-home message here, is that the Andropogonii genomes are all similar in gene content, and the architecture is also very similar. That is, they line up really pretty well, except for all the species of Zea, so not just maize, but the wild ones as well, in which the genomes they're collinear within Zea, but extensively rearranged relative to the rest of the tribe. So after a fairly long presentation, I'll sort of return to that conclusion. So step one, what does it take to assemble the germplasm? Well, it takes a bunch of people in the lab, a bunch of really good horticultural staff, excellent greenhouses, multiple collaborators and collectors, several dozen land managers, colleagues at 3 major herbaria, and a whole lot of collecting permits, because many of these things grow. If you want adapted germplasm, then you're looking in areas that are native grasslands, which are actually endangered in many parts of the world, as well as international permits. So a lot of time just requesting permits to collect. The team at the Danforth Center, I've already mentioned the folks, I think, in few folks in my lab. Taylor Aubuchon Elder is the person who really has led and organized this project. She's the senior lab technician in the lab, working with Pat Minks and Bess Bookout. And then the lead in the horticulturalist in the growth facility, Sally Fabry, was absolutely critical, and her assistants, Stephen and Olive, and then a bunch of collecting with our colleague Christine McAllister at Principia College in Illinois. So it's not a huge team, but everybody was critical to the whole operation. We started with a list of species in the tribe. So this is just under 1,200. It's about 1,174 or something, starting with several online databases from the Royal Botanic Gardens at Kew. So grass base, and I put up a picture of Derek Clayton, who passed away just recently, and Plants of the World Online, and our other collaborator at Kew, Bat Vorontsova, and getting lists from books. From these lists, we can find localities where the plants have been collected in the past, so that's how we know where they grow. We can get general aspects of the morphology and some bits of the life history that are most are perennial, long-lived, but there are a few annuals. What we can't, back up. Species names, just to remind you, are based almost entirely on morphology, not experimental work. It's people working in a herbarium, looking at things and saying, golly, that looks different, and giving it a name. And of course, for a project like this, from the lists of names, we can't find genome sizes, breeding system, germination requirements, growth requirements, environmental conditions, physiology. The only bit of physiology we can get is the fact that they're all C4. And we can get that just because you can just figure it out by looking at the plant. You don't need to do an experiment. So the whole group went out collecting, starting the minute the grant was funded. And we got live plants, silica dried leaves, and herbarium material. Live plants are by far the best, and I'll go through just a little bit about that. Silica dried leaves are okay and have some advantages, and herbarium material is kind of the last resort, but it works. So living plants are good. You probably never thought about there being any alternatives. So they produce good quality long strand DNA. They can be used to estimate genome size and count chromosomes. Can be used for RNA, propagated clonally, so you can pass them around, and they're available for experimental work. Some stuff we can get from seed banks and germplasm collections. So you might be familiar with the USDA germplasm network. There's a seed bank, a substantial seed bank at the Royal Botanic Garden at Kew. And then Pat Minks turned out to be really good at finding stuff on eBay. So those things work. It's great. You can get diverse germplasm from places that are hard to visit, and the seeds are really easy to get around. The downside is that the identifications are often wrong. So the staff at these seed banks do not have time to grow up and validate the identification. So you need to grow them up to flowering to check the ID. The other thing is these things are not inbred. They're usually bulk collections across multiple plants, so that there's variability with what's in the packet. You don't always know about germinability. If the plant is annual, you need to keep propagating it, and there's often very limited information on the source locality. So using that resource, we got 60 species out of our 1,174. So that was a good start. The next step, of course, was field collections. And that's, you just go get them. Use maps, herbarium records, natural area species lists, buy a plane ticket, dig up the plant, make a voucher specimen, about which more in a second, cut back the leaves, put it in a Ziploc bag, bring it home, stick it in a pot, and quarantine it to let the bugs leave town. Now I get asked frequently, well, why not collect seed? And the answer is that the timing is really tricky. You have to get there exactly when the seed is mature but hasn't fallen off the plant. And of course, with wild plants, they don't hang on to their seeds. The other problem is that you usually don't know how to break the dormancy or make the things germinate. And so, although collecting the seed may be fast, it doesn't save any time when you get the thing home. So that puts a premium on getting good locality information, and Taylor retrieved this from GBIF, the Global Bioinformatics Information Facility, and BN, which is a network that basically cleans up a lot of the GBIF data and deals with some of the errors, the known errors in GBIF. In this map, each dot is one plant, so this is where we have records of specimens of Andropogonii. There are some artifacts here. For instance, it looks like there's not much in India, and the reason is it's a data entry problem. The Indian taxonomy community has not made as much progress as some other parts of the world in getting their data into these global facilities. So there are actually quite a lot of species in India, but they're currently somewhat inaccessible. Yeah, that's basically, and just as a reminder, of course, GPS has only been available since 1978. People have been collecting plants and putting them in herbaria starting in about the mid-1500s, with the major activity really speeding up in the 1700s. So you've got quite a lot of old specimens available, but of course, somebody who was collecting in like 1820 was not putting down latitude and longitude. So here's just in the background a picture of these guys in the greenhouse. They're perennial, they're wild, they're not inbred, so they're heterozygous. We don't grow them for seed also because they're heterozygous, so the plant is kind of what we've got, and so we just propagate them in the greenhouse, and hence my reference to the tremendous greenhouse staff that we work with. I have mentioned a voucher specimen, so this is kind of a public service announcement for people who do field work or work with non-domesticated plants. A voucher specimen is a pressed plant, and it provides an identifiable record of what you actually got, never mind what you thought it was. So when you collect the plant for DNA or bringing it home, you press the specimen between two pieces of paper, write the information in a notebook, assign a unique identifier, send it home, type up a label. So you can see in this specimen here, there's a label here with a bunch of metadata on it, and you deposit it in a herbarium parser at the Missouri Botanical Garden. And this constitutes a permanent record. So that means if I say, well, I collected, I don't know, big blue stem, and then something funny comes up with the data, I can always go back to that specimen and say, well, did you identify it right? Or pick up something peculiar by mistake. So this allows validation of the identification. I also often get asked, why not just take a picture? And the reason is that in grasses, the diagnostic characteristics are often tiny. They come down to the florets, which are just a few millimeters long. And with a photo of a specimen like this one I'm showing you, you really can't tell what you've got. I know it's a grass, and I can guess it's probably Hyperrhenia, but based on the inflorescence, but I couldn't get it to species from that. So the specimen is the thing that lets you go back and really, because you can dissect these things after the fact. So that necessary detail. All right, so with live plant material field collections, you've got all the advantage of live plants in general, but they're hard to maintain. They take up space. The greenhouse is expensive. We do pay greenhouse fees, so we pay for this by the square foot. They attract bugs, so they need really good care and need to be continually regenerated. Mentioned the permits, and particularly if there's international, then you're dealing with international shipping. There is another hazard, which is contamination, and we did run into this in this project. And if you look at this well-known painting from Dürer in 1503, this picture of turf, and the grass here is Poa pratensis. It's what we would call Kentucky bluegrass, you can just see that all the roots of these plants are all sort of intertwined. And so if you go out with a shovel and dig this up, you get mixed collections at the start, and you then have to do your best to try to keep things cleaned up. We did that, but nonetheless ended up with a contaminant in one of our, what we thought was going to be a reference plants. It was supposed to be big blue stem. This thing, as you can see, is labeled a weed. It turned up in the pot, and we sequenced it by mistake. Spent a lot of time sort of agonizing about what had gone wrong, but ultimately it turned out it's a species of POA. The thing, I just finally did a freehand cross section of it, and it's clearly a C3 grass, which you can see by the spacing of the veins, which means it's not Andropogonea. There's no question that we got the wrong thing came up in the pot. This particular one turned out to be kind of as Alyssa Phillips coined the title, a happy accident. There was no genome for Poa pratensis. This is Kentucky bluegrass, which is widely used horticulturally. And Alyssa and Arun Seetharam on the right, put together the genome and managed to put together the paper and a lot of other information that was necessary to publish this. And despite its economic importance, there is no other published genome for this species. So in this particular case, although we initially thought it was a disaster and a colossal waste of sequencing money, thanks to Arun's ability to separate the weeds from the POA out of the Andropogonea, out of the Andropogon reeds, and then Alyssa's ability to do further analyses with it, it turned out pretty well. So occasionally mistakes pay off, but I don't recommend it. It still makes sense to avoid contaminants. So, okay, so now we've got live plants. We got 136 species represented by live plants. So what's the next choice? Well, silica dried leaves do work pretty well. I would put them in as okay. The trick is you go down to a craft store, don't buy it from Sigma because it's too expensive. You get silica gel for dried flower arrangements where you can get it very cheaply. Put a little bit in a Ziploc bag, take the leaves off the plant you're interested in, put it into some sort of porous material, which might be a tea bag with the tea removed or a coffee filter or a coin envelope. Put the thing into the Ziploc, and the thing, you've effectively lyophilized the leaf. And then, of course, you make a voucher specimen. The good thing about this is that it's easy and cheap, easy to transport, easy to store, good quality DNA, and colleagues can send material easily. And this is a really important way of extending your reach. Of course, the downside is you don't have the plant. So you can't do all the things that you would normally do with a live plant. So we had many colleagues sending us silica material, and I've just posted a few of them here. Cassiano from Brazil, Remy Pasquet and his colleagues from Africa. Again, Bat Vorontsova from Madagascar and Wachara Arthan from Thailand. A way of getting things from around the world. It works pretty well. And then finally, when all else fails, or when there's a global pandemic and nobody can go anywhere, you then resort to the herbarium. It's straightforward. You find a specimen with enough leaves, remove a little bit, you annotate the specimen to show that you've just destroyed a small piece of it. So you just glue a little label on, like material removed for DNA extraction. And obviously the really good thing about this is you can get a huge sample of species. People have been collecting this, as I said, for a couple of 100 years, so you've got a lot of choices. The DNA may be okay. You don't need permits. Of course, the downside is the DNA is often degraded and you usually don't know ahead of time. And it still requires some travel, and you don't always know ahead of time what the herbarium is going to have because of the database issues. And of course, then we went back to a bunch of stuff that we had in the freezer because that's what you do. And we have a lot. So the total, and this is, as I said, Taylor's driving force and organization behind this, was 750 species sent for sequencing. These are the different kinds of DNA shown here on the left, and they don't all quite add up to 750 because there's a certain amount of duplication. So locations are roughly shown here, except in these ovals where there are too many collections to get them on the map and still read it. For current analyses, which I'll get to in a minute, working with a little under half of those things. So it's about 25% of the tribe, which is still a lot. As an aside, just one mention of the population analyses. We don't have results from this yet, but for 10 species, we also sampled population level, 10 plants per population, 10 population. So this is about another 1,000 plants, and Pat Mink's estimated genome sizes for every single one of them. We kept them all alive for several years. But I'm not going to talk about the population work any further, just to say that that was part of what we did. So, so that then gets on to DNA sequencing. Got all the stuff, now you sequence it. And the long read technology focused on 35 species, mostly PacBio Hi-Fi, some nanopore. And the assemblies and annotation were led by Arun Sitharam in Matt Hufford's lab at Iowa State. And then everything else got sequenced with Illumina, handled expertly by Cinta Rome, who is the project manager for this whole project, working at Cornell. So she was the one, we sent the material to her, and then her team then dealt with the sequencing. And obviously assembly as well. And although this is a ton of work, I'm only going to give it one slide in this particular talk. It's all de novo. So there's no resequencing here. And the reason is, of course, that if you want to look at presence absence variation, the resequencing doesn't do it. And again, for all these guys, Pat did the flow cytometry. So it could be a guide for the assemblies. And for the things with long read sequences, we also collaborated with Patrice Albert and Jim Birchler's lab at University of Missouri. And she did some chromosome squashes. So for a small number of these things, we actually have not just genome size, but real chromosome counts. So here are two of the diploids with dramatically different chromosome sizes. All right, so I've told you about the first 3 bullet points on this list. So I'm going to jump ahead briefly, and that is the comment about the phylogeny, because I need to bring in some information from that to be able to chat about the other tools under development. So here's a sort of a sparse phylogeny. We have various size phylogenies that various people have built. I just chose this one because it shows what I needed to show. It's most of those, the phylogenies are, the big patterns are largely in agreement with published phylogenies and additional ones that we've gotten. So the points that I'll ask you to sort of pay attention to for this talk is that the things on that obvious long branch right there that's all the species of Zea. So that's one clade, and it is sister to the genus Tripsicum, which may or may not be as familiar. Most of you, of course, are wondering where sorghum is. It's right there. So it's in the tribe, but it is not close to Zea. And then for the talk today, the rest of it doesn't matter in detail. There's just a lot of it. So here's Trypsicum. Just if you haven't looked at it, it looks, kind of like teosinte, with pistillate spikelets at the bottom, and paired staminate spikelets at the top. And this is just the pistillate spikelets, and the fruits are like, they're not dissimilar to teosinte, but softer, not quite as rock-like. All right, so with that very brief phylogenetic excursion, then tools for improved annotation of the maize genome. And this is work from Amy Schultz and Shenkai Xu. And this was uploaded to bioRxiv last night. So brand new stuff. And they assembled a pipeline they call RealGene for using cross-species comparisons to improve the maize annotation. And thanks to Amy for sharing a lot of these slides. So if you look at the different annotations of the maize genome, for some loci, many loci, you get alternative gene models for what should be the same locus with different intron-exon structures and different UTR boundaries. And the goal here with this tool was to use all the sequences across the taxa that I've just described and use that to help evaluate the models in maize and use the conservation as another clue as to where starts and stop codons and intron-exon junctions are. So the general procedure here is to take a transcript, a maize transcript, and align it to all the Andropogoni assemblies. In fact, in this particular project, ended up with 91 of them. And this is work of Zach and Brandon at Cornell. So align it to everything and get a multiple sequence alignment for each transcript. So now across taxa. And then develop models using various machine learning processes to evaluate and come up with criteria for evaluating the likelihood that the maize model is going to be functional, that it's actually going to work. So there's one model which is based on UTRs, basically looking at transcription starts and transcription stops. Another based on exon intron boundaries, and a third one based on proteins. And using evolutionary conservation, then this predicts the UTR boundaries using a bunch of machine learning processes and getting to functional transcript boundaries. It also predicts key molecular junctions, like translation starts and stops, intron boundaries. And the protein model predicts functionally conserved proteins in maize. This is work of Jingjing Zhai. And then combining those three models, Amy and Shenkai and Jingjing were able to generate a score. And that score was then used to estimate whether a particular gene model was likely to be functional or not. So just to skip over a whole lot of work and some really intriguing methods, they started with classical genes. They decided, obviously, you need a positive control here to know if you're right. So they started with ones that had at least 3 publications and real good data on gene structure. So the things that everybody would any reasonable person would agree is really a gene and really annotated properly. And their combined models predicted that 100% of those things really were functional, which is good because that's what was expected. Large percentage of genes in the maize proteome, large percentage of genes in the transcriptomes, and then a much smaller percentage of predicted genes that were not in one of these categories. The most intriguing thing from an evolutionary point of view was looking at comparisons of genes across species. So for these, if they had gene models that were shared outside of Zia, most of those were predicted to be functional, to encode a functional protein and to be annotated properly. Within Zia, there are a whole lot of things that appear to be Zia specific and a much smaller set of things that appear to be maize specific. Superficially, you would think these are the things that make Zia or make maize what it is. They're, evolutionarily important, encoding novel functions of some sort. However, very few of them appear to be functional. So 2.8% of the ones that are unique to Zia, 2.4% of the ones that are unique to maize, which suggests that an awful lot of those maize-specific predicted genes are either pseudogenes or they're just misannotations. The flip side of that, of course, is having set those aside, you then end up with a very small but potentially very important subset that might be worth much more investigation. So the summary of these results is just the combination of models improves the annotation of the maize genome, and the cross-species comparisons, like all that germplasm, all those sequences, are actually integral to these models and help improve the results. The computational pipeline should work for any species for which sufficient cross-species data exists. So this may be a very general tool that's just been validated in maize. And as I said at the beginning, there's going to be a DOI in the next few days, hopefully. So it'll be pretty exciting to see this moving forward. Right. So that's improved annotation of the maize genome, which is useful. What else do we need for tools? Well, you need to be able to align whole genomes that after Arun has generated all these lovely assemblies, and we pull some nice assemblies out of the literature, how do you line up everything across the genomes? And obviously the issue is that the genes are going to align fine. You can find orthogroups. That's not a problem. But the space between them creates problems, particularly if you're interested in what's happening in the space between them in terms of regulatory sequences or transposable elements. And the other issue that I haven't mentioned much, but is a serious one in this group, is polyploidy. It's very, very common in this tribe. There are, of course, dispersed repeats, and there are areas where there's a lot of sequence diversity in that intergenic space. So one tool that's been available for a couple of years now is Anchor Wave, which was developed by Baojing Song and Michelle Stitzer. Baojing has his own lab now, but and they're both in Ed's lab when this was developed. And Michelle kindly shared her slides. The genomes are hard to align because of these repeated sequences. And I think I probably don't need to tell this audience about transposable elements in maize. But as you know, they're genes, and then they're transposable elements that are sort of speckled all across the genome in between them in varying contents. So one way of dealing with all those pesky transposable elements is to pretend they're not there, mask them, and then focus on the gene, which gets certain answers, but doesn't really address the question of how that intergenic space is changing. That is, you can go ahead and align those genes. But what the anchor wave program, Baojing and Michel's program, does is uses the genes as anchors. So it aligns the genes, and then it separates out the space between the genes and aligns them separately. So you've got the genes, the blue gene and the orange gene aligned. And then you've got the TEs that are in between them. And then you've got this other gene and orange gene aligned here. And now you create basically it becomes a separate problem to align the space in between them and analyze that variation. And it can also account for old polyploids by specifying how many, whether you're looking at a 1-to-1 arrangement between the genes or a 1-to-2 or 3, depending on what the polyploidy level is. So in this particular case, it's 2 syntenic blocks after a genome duplication. So the orange gene, the entire orange genome is duplicated in this diagram. So using that particular algorithm, then, Michel has taken Arun's assemblies and started aligning them. And I've gone back to the phylogeny and just stuck stars on as to where we have the good long read assemblies. Not to discount the short read stuff, but for the moment, I'm talking about the long read assemblies. And these were chosen for very dense sampling in Zea and its sister genus Tripsicum. Obviously, sorghum is in the literature. And then the others were chosen to capture the phylogenetic diversity of the tribe. So specifically chosen for their phylogenetic position. And I need to add in just a little bit more information here into this. I assume you all know that Zea has a haploid chromosome number 10. And it turns out everything else in the tribe, most other things in the tribe have a haploid chromosome number of 10 as well. So all these guys up here, all these guys down here. So we're looking at 10 and 10. And we'll come back to that in just a second. So if we just look at all those, here are alignments that Michel put together for the long read sequences. Very large blocks of collinearity. They're aligned against Paspalum vaginatum, which is not Interpagani. It's an outgroup, which also has 10 chromosomes. And you can see that there's some discrepancy, some variation around Paspalum chromosome 5. There are a few places where it looks as though there's missing sequence in a couple of species. It's not clear that that's missing. It may not be aligned very well. But in general, there's a lot of similarity across these genomes, including similarity with the outgroup, suggesting it's persistent throughout the evolution of the tribe. So the genome organization is conserved among all these X equals 10 species, whether diploid or polyploid, except Zea, where we know there are a whole lot of rearrangements that have been studied by a lot of people for a long time. And of course, the other bit of information that most of you probably know is that there's a whole genome duplication in the common ancestor of Zea and Tripsicum. And so all these rearrangements in Zea have often been rearrangements and fractionation, often attributed to this whole genome duplication, which then led directly to the rearrangements. And of course, if you look at this phylogeny, the key to that in answering seeing if that is actually true requires looking at trypsicum, because trypsicum is also a descendant of that duplication. The chromosome, haploid chromosome number in trypsicum is 18. It's not 10. So here it is again, 2n equals 18. And so Michelle lined this up against Andropogon virginicus, which is one of those x equals 10, the non-Zia x equals 10. And what you can see is that, like, chromosome 1 in Andropogon virginicus maps pretty well against 2 chromosomes in trypsicum. Likewise with chromosome 2, everything maps to 2 places. Chromosome 3, things are a little scrambled in 4, 5, and 6, but there's still a lot of collinearity, and then these other ones all, you know, it's a 1 to 2 mapping, consistent with the whole genome duplication, except we're looking at 18 chromosomes. And that suggests a model like this, which has been a possibility for some time, but this is the first evidence that it might actually be more than a possibility. It might be real. So you've got a 10 chromosome haploid number, 10 chromosome ancestor, which I put in blue, another one which I put in orange, and it might be an 8 or a 9. We don't know that, but those things obviously hybridized. There was a whole genome duplication. Just basic arithmetic says the immediate product of that whole genome duplication should have been something with a haploid number of 20. 2 of the chromosomes obviously got rearranged at that point, leading then to trypsicum with an n equals 18. And then all the rearrangements in Zia happened on the way to Zia, but not back here, or not immediately after the whole genome duplication. That is because these things come from the same allopolyploidization event, because trypsicum is super similar to the rest of the tribe. The architecture of the Zia genome is a downstream, I mean, it may be a downstream result of polyploidization, but it probably wasn't the first thing to happen, or we would see it in trypsicum if it were. So I think I can go for another 2 minutes. So I will mention the transposable elements, because once you get these alignments, you can start looking at them. So this is another of Michelle's diagrams, where each bubble represents the proportion of the genome represented by that particular transposable element. So the rows are the species that had long-read sequences, the individual plants that had long-read sequences. This histogram here is the assembly size, and then each column of bubbles is a different class of transposable elements. And just by eyeballing this plot, you can see that there's just a lot of similarity across most of the tribe for most of the transposable element classes. That's not without variation, but it's pretty similar. And I put boxes around the rows representing Zia and the rows representing trypsicum. So those are the guys after this whole genome duplication. And a couple of striking things came out of this plot. One is that one whole class, these are the TC1 mariners, are nearly gone after the whole genome duplication. Don't know whether it's causative, but you can certainly see the pattern. And then there's a helitron expansion, which is shared by all members of Zia, but only Zia, not with trypsicum. So that's clearly, so this loss of the mariners probably occurred very soon after the whole genome duplication. The helitron expansion was later, coinciding perhaps with the rearrangement of the Zia genome. So, all right, so we've done some stuff on identifying conserved non-coding sequences. And I'm only going to give this one slide partly because this is still work in progress, but Baojing did some work on this a few years ago, basically showing that conserved non-coding sequences that were conserved across the entire tribe were harder and harder to identify. There were fewer and fewer of them as you looked at more and more species. Armin Shaban did a similar analysis on shared transcription factor binding sites. And again, the more species you included, the less conservation you'd find across the whole group. This isn't too surprising. And Armin has done a number of analyses to sort of take apart this pattern, because this is obscuring a whole lot of nuances of particular sorts of sequences and particular patterns of evolution. But that work is ongoing. So I threw in a few slides on biogeography, but I think I will skip those in the interest of time. Right. The maize and Tripsicum group is this green dispersal to the New World, and we don't really know where the ancestor was. So, right, and just conclude here. Then the genome structure I've been talking about is at least 20 million years old. And in general, this largely collinear genome structure has persisted and has spread everywhere in the world, has indeed taken over the world. And the maize genome has a lot of similarities to the other Andropogoni, but the genome arrangement seems to be Zea specific. And that too has spread all over the world. So I'll stop there. And if there are any questions, happy to take them for a few minutes, and then I've got another meeting with some of you in about 8 minutes. So
thank you very much, Toby. I have a couple questions in the chat. Other folks, please feel free to put questions in chat for Toby. I'll skip the one on silica gel in the interest of time. The arrangements you showed on Tripsicum and Zea, the polyploidization event, so the 2 chromosome arrangement, that happened before Zea Tripsicum split?
Mm-hmm.
And then do we have any better idea of whether the polyploidization event for Tripsicum Zea was segmental allopolyploidy? And maybe if you could, I'm always confused about exactly what segmental allopolyploidy is.
Yeah.
You could comment on that.
Yeah.
Yeah.
It sometimes seems to be more a description of a pattern than an actual mechanism.
Right.
Yeah, I don't know, is Michelle here? She's looked more closely at this genome structures, and I think she'd have a better answer to this than I would.
Yeah, we don't have the evidence yet, but I'm in the process of looking for it. So segmental allopolyploidy is going to be when, essentially when polyploidy occurs, you can either have those duplicated chromosomes segregating truly as diploids, or those 4 chromosomes can be equivalent to one another and pair in different ways. And so if those are segregating as quadrivalents, or if all 4 are interchangeable, you get kind of different segregation patterns and different recombination patterns, because then those can recombine with one another. And so segmental allopolyploidy is the idea that some of the chromosome pairs are segregating immediately as diploids once they get into that polyploid genome nucleus, but that some of them have this longer period where they can kind of interchange with one another and sort of trade. And so that changes a lot of how effective selection is going to be, all of these sorts of things. And that pattern maybe should be possible to see in whole genome data, but we're still in the process of kind of thinking through and looking at that.
Well, and I think it's going to give the same pattern as homologous conversion of genes, and then you'd be starting to worry about dating and timing and things. And so I think that the segmental allopolyploidy or not, to me, seems like a difficult question. I mean, that is, you can presumably identify regions where things do not sort neatly into two homologs. exactly what did that and when is, I think, a harder question to ask. Well, it's not a hard question to ask. It's a hard question to answer.
Yeah.
And then Ruben, you had a question on chat. You want to unmute?
Yeah, it's a very basic question. Why do you think that the SIA went through much more rearrangement than Trypsicum? What's in the biology or the reproductive system? Do you have any intuition about that?
The reproductive system is pretty similar, because they're both monoecious. It's the architecture of the plant is, I mean, Trypsicums are pretty robust perennials. Most Zias are annuals, but I think as Amy Schultz has been working on this annual perennial business as to sort of when and where that turns up in the phylogeny. And I think, I don't know if she's here, but she may be sleeping since she got that manuscript submitted last night. Oh, there she is. She's awake. Yeah, congratulations, Amy. Anyway, yeah, Amy, maybe, do you have some thoughts?
Yeah, I guess I'm diving into this more, and I haven't looked specifically into the timing just yet. But it looks like it's pretty dispersed across the tree where annual and perennial occurs. So, one of the questions I do want to ask is related to potentially what that environment, if there's an environmental driver or other, timing positions of it. But that's a great question.
Yeah, I think that, I mean, Ruben, the straight answer is, I don't know. I don't think I mean, the annual perennial is an, an obvious possibility, but I don't know what controls genome rearrangements in general. That there's some things where the genome rearrangements are common and actually frequent, and then there are other clades where it seems to be absolutely fixed. And I just don't know what the answer is. It's a great question. Absolutely great question.
Thank you. Great work.
We're nearing the end of the hour. Unless there are any other questions in chat, let's thank Toby again, and we'll be back next week with a talk by Ellie Cryan. So tune in next week, same bat channel, bat time, etc. Thanks, folks.
Great. Thanks, everybody.